All right, today we are cracking a case. We're diving deep, I mean really deep, inside our own cells to solve a kind of molecular mystery. How in the world do you find one tiny, specific secret hidden inside our massive genetic code?
And the key to this whole thing really comes down to one incredible question. How is it even possible to paint a chromosome? I mean, it sounds like science fiction, doesn't it? But stick with me, because the way scientists pull this off is nothing short of brilliant.
So, first things first, let's lay out the crime scene, so to speak. The central problem for scientists has always been this. How do you find a single specific gene, or maybe a piece of RNA, in the middle of the gigantic, incredibly complex library that is our DNA?
To really get a handle on this, think about it like this. Trying to find that one gene is like trying to find one specific sentence somewhere in an entire library of books. The scale of this problem is it's immense. But luckily, science has a master detective up its sleeve.
And that detective is a technique called In-situ Hybridization, or ISH for short. It's this super powerful method that lets scientists find and then literally light up a specific bit of genetic code right where it lives. That's what in situ means, in its original place. We can do all this without ever having to rip the cell apart.
Now, every good detective story needs a team, right? And for this investigation, our team has two main players: the target we're looking for, and this amazing high-tech tracker we send in to find it, which is called the probe.
Here's the easiest way to think about it. The gene or RNA we want to find, that's our suspect. The cell it's hiding in, that's the city. And our labeled probe, well, that's like a highly specialized GPS tracker, one that's been engineered to find that one suspect and completely ignore everything else.
So what exactly is this probe? Well, it's a little piece of DNA or RNA that scientists custom make in the lab. And here's the clever part. They design it to be the perfect, complementary mirror image of our target. You know how in DNA, A always pairs with T and C always pairs with G? Because of that rule, this probe is basically a heat-seeking missile. It'll ignore millions of other sequences and lock on only to its perfect match. It is the ultimate specialist.
So we've got our target, and we've got our probe. How does the actual mission unfold? Well, it's a very precise operation to catch our genetic suspect, and it happens in a few key steps. First, you have to prepare the scene. The cell is preserved, but also made a little bit permeable so our probes can get inside. Then, step 2, send in the probe. Thousands of these little trackers are released to hunt for their one and only target. Step 3 is absolutely crucial. Wash away all the distractions. Any probes that didn't find a perfect match just get rinsed out. And finally, step 4, reveal the location. This is where we use a special method to see exactly where our probe, and therefore our target, is hiding.
Okay, but hold on. How do we actually see the probe once it's found the target? I mean, a successful mission is pretty useless if the detective can't signal back to tell us where they are. And that's where the probe's label comes into play.
This label, or reporter molecule, is basically a tiny chemical tag that gets attached to the probe before the whole mission even starts. It doesn't get in the way of the search, but it's the thing we can actually see at the end. It's literally the detective's flashlight in a dark room.
Now, back in the day, these labels used to be radioactive. They were incredibly sensitive, which is awesome for finding targets that are really rare. But they're also hazardous to work with and took a long time to process. These days, non-radioactive labels are way more common. They're just safer, faster, more stable, and over the years, their sensitivity has gotten so much better. And the real superstar of the non-radioactive world is fluorescence. When the labels are basically tiny fluorescent dyes, the technique gets its own special name: fluorescent in-situ hybridization, or, you guessed it, FISH. And this is where things start to get really, really colorful.
So look, this is way more than just a cool lab trick. In the real world, ISH and FISH are absolutely essential tools that have helped crack some major cases in both medicine and biology. For doctors, ISH is a frontline diagnostic tool. It can spot the DNA of viruses like HPV or hepatitis B hiding right inside our cells. It's also critical for cancer. For example, a FISH test can tell doctors exactly how many copies of the HER2 gene are in a breast cancer cell, and that information directly tells them what treatment to use. It can even identify specific bacteria in a sample without having to do the slow process of growing them in a dish.
And beyond medicine, ISH is just fundamental to helping us understand the blueprint of life itself. Researchers use it to see which genes are getting turned on or off as an embryo develops. It's been used to create these incredibly detailed maps of where our genes sit on our chromosomes. And it can spot big genetic problems like when parts of chromosomes are missing or when pieces from two different chromosomes get swapped by accident.
And the sheer power of this it's breathtaking. The sensitivity is so precise that it can reliably detect as few as 10 to 20 copies of a target molecule inside one single cell. Think about that. That's like finding a single grain of sand on an entire beach. It's just amazing.
So all of this brings us right back to our very first question. Now that we know we can design a probe for pretty much any gene, and we can label these probes with all sorts of different colors, we can finally understand how to paint a chromosome.
Because what you're seeing in these images isn't just a pretty picture. It's so much more than that. It's a precise, color-coded map of our genetic instructions made visible to the human eye for the very first time.
The technique is called multiplex FISH, or M-FISH. And what scientists do is they create this incredible cocktail of probes. Probes for each of our 23 pairs of chromosomes get tagged with a unique combination of fluorescent colors. When you apply that cocktail to a cell, each chromosome grabs its specific probes and lights up with its own unique color signature. We're not just finding one sentence anymore. We're lighting up the entire library and giving each book its own colored cover.
This has completely revolutionized genetics. It allows scientists to spot really complex chromosome damage, like in cancer cells, with just a single glance. And it just leaves us with this one final, really exciting question. Now that we have the power to light up any gene, anywhere, at any time, what mysteries inside the cell are we going to solve next?